Hello everyone and welcome to the next video. This video is part 2 of photosynthesis. So in part 1 we discussed about the light dependent reactions which made ATP and NADPH for us, and in this video we will be discussing the next phase, phase 2 of photosynthesis, which is also called the Calvin cycle. And Calvin cycle, please remember, they're all light independent reactions, okay? They don't depend on light. So as we will just uh, revise what we did in the last video, we made NADPH and ATP after phase 1 of photosynthesis. It's not stable enough to store your energy for a longer period of time. So the plants will try to store them in organic molecules such as glucose, which is your final product of photosynthesis. We know that. So coming to the Calvin cycle, where does the Calvin cycle takes place? It takes place in the stroma of the chloroplast. We already discussed this in the last video. Remember, I'm not discussing the Calvin cycle in details. Just our basic knowledge will do. Okay. So Calvin cycle is divided into 3 phases. You have carbon fixation, reduction, and regeneration. So in the first phase, which is carbon fixation, it's very simple. You have a molecule called RUBP, ribulose 1,5 bisphosphate. So this 3 molecules of RUBP will combine with 3 molecules of carbon. So I have 5 carbon here and I have 1 carbon here. So 5 plus 1, 6 carbon. So it will make a 6 carbon product called your something here. I'm not gonna go into details of it. A 6 carbon molecule which will break or split into 2 3 carbon molecules, which is called a 3-PGA. These details are not required. Just remember, what is happening in the Calvin cycle. What is important is Rubisco. Rubisco is an enzyme which helps in this carbon fixation. Okay. So this particular reaction is catalyzed by an enzyme called Rubisco. The next step is reduction. So you made 2 molecules of your PGA. So what you will do next is you go on converting from one molecule to the other. So in this process, you make use of 6 ATP. So you're not making ATP here. You are using the ATP which you made in phase 1 to make ADP, and you're making use of NADPH which you made in phase 1 to make something called NADPP+. Okay. Now, so out of all this, you get here 6 molecules of your 3-phosphoglycerate, and the final product here is G3-phosphate. Okay, out of 6, 5 will continue in the cycle and 1 will go to make glucose. Please understand, because in the next step what happens is out of the 6 product that you are making, which is G3-phosphate, 5 will go back to the cycle. Only 1 will go out of the cycle to make glucose for you. And what is this going back called? It's called regeneration. So this 5 G3-phosphate will go to make your back to the first compound IUBP, and this cycle continues. So this is what the cycle is. You start from here and you go back to IUBP. In this process, only 1 G3P goes out of the cycle to make glucose. Okay. Next is, this is the process, the photosynthesis that we discussed is for the normal plants, which are C3 plants. Okay. There are some plants which live in extreme climates. Okay. Those plants are of 2 types, C4 plants and CAM plants. C4 plants are those plants which live in conditions where they have to minimize water loss. Okay. So they occur in certain plants like sugarcane and corn. So here the C4 plants, they fix the carbon dioxide not in a 3 carbon molecule like just now we saw. What are we doing? We're fixing the carbon dioxide in a 3 carbon molecule in the Calvin cycle, right? But here for C4 plants, they don't fix it in 3 carbon molecule compound. They do so in 4 carbon thing. Okay, that's why their name is C4 and not C3. So they fix the carbon in a 4 carbon molecule compound. Okay, what is needed here is, here they also have the light dependent and the light independent reactions, but here these two reactions are separated in different parts of the leaves. So the light dependent takes place in the mesophyll of the leaves, and the Calvin cycle takes place in the bundle cells of the leaf. Okay, so these plants, they are found in hot climates. That is why the whole process of C4 pathway is to minimize the water loss. Okay, the next thing is CAM. This also takes place in somewhere where it is dry. So this one for hot climate, and CAM plants are usually for the plants which live in desert or in very dry areas. Okay, so here also the purpose is to conserve water, but here instead of having the light dependent and light independent reactions in different places in the leaf, they don't do that. So this is when you separate the plants with respect to space. Okay, different parts. Here we separate the plant with respect to time. So one part, like say the light dependent reaction will take place during the daytime, and the light independent will take place in the night time. Okay, so this is what you have here, the table. In C3, the normal plants, there's no separation. Your light independent and dependent both takes place in the same part of the leaf, takes place in the daytime. It's for cool and wet environments. C4 plants is when the things get separated in space. So the light dependent reaction takes place in the mesophyll of the leaves. The light independent reaction takes place in the bundle cells of the leaf. It's best adapted to plants which live in hot sunny environments. CAM plants are those which live in very hot dry environments. So there your light dependent and light independent is divided based on time, not based on space. Okay, the last thing that you have to know here is the factors affecting the rate of photosynthesis, which is a very important topic again. The two important factors which affect the rate of photosynthesis is your light intensity and carbon dioxide concentration. How does light affect the rate of photosynthesis? So here, as you start increasing the intensity of light, photosynthesis also increases till a particular level. At very high light intensities, you see that photosynthesis is not increasing, it's slowing down, and gradually it's totally inhibited after that. Why it's inhibited? Because the various factors that this thing happens. Okay, I'm not going to the details of this here. Please remember, the photosynthesis will increase till a particular light intensity. After that it will start decreasing, and finally it will stop. Same goes with carbon dioxide concentration. As you try increasing the carbon dioxide concentration, photosynthesis rate will increase for some time. After that, then you will see that at some point the photosynthesis will either be stopping, or they will slow down at some point. Okay, even if you increase the carbon dioxide concentration, the rate of photosynthesis will not increase. Now finally, before we wind up the video, let's practice some questions. So here in this question, they give you a graph which shows the effect of light intensity and carbon dioxide concentration both in one graph. Remember that, okay, on the rate of photosynthesis. Now they ask you, so here look at the concentration. Here the concentration of carbon dioxide is less. Here the concentration of carbon dioxide is more, right? So we know that as the concentration of carbon dioxide increases, photosynthesis is also increasing to a particular level. After that, no. And even for the light, see, this is your light intensity on the x-axis here. So as you go increase the light till a particular level, photosynthesis will increase and then it'll stop. So let's look at the options that we have. Carbon dioxide is not a limiting factor for photosynthesis, is it? No, this one is wrong. Carbon dioxide is a limiting factor. Because what is a limiting factor? That you increase, when it keeps on increasing the rate of photosynthesis, after some time it will stop it. Okay, so that's a limiting factor. It limits the rate of photosynthesis. Now, the next thing is photosynthesis rate is proportional to light intensity, which means as you increase the light intensity, photosynthesis will always increase. Is that the case? No. Photosynthesis rate will increase only till a particular level of light intensity. After that, it will not increase. So we cannot say it's totally proportional, right? Now, the next option is the rate of photosynthesis is limited by carbon dioxide only at low light intensities. Not necessary. Look at the video, look at the graph here. At low light intensities, it's not limited by carbon dioxide, still increasing. Okay, so it doesn't make sense. Look at the last option. Light intensity and carbon dioxide both limit the rate of photosynthesis. Yes, they do. Because we know after a certain light intensity, after a certain carbon dioxide concentration, your photosynthesis rate will stop or it will be affected, right? So your answer would be the last one. With this, we end the video. And in the next video, we'll be discussing a totally new topic, which will basically be your light microscope, and to compare it with electron microscope. It's an easy unit, but we will try to talk about that. Thank you so much.